Hi everyone, today I'm going to share some information related to biosafety and particularly in a research setting. I'm Christina Yong, I'm a researcher and a lecturer from the Department of Biology, Faculty of Science, and I'm also a member of the Biosafety Committee, UPM. Let's start our talk with this. An incident that is very current and relevant to the topic today as well as our life nowadays. The COVID-19 pandemic caused by the SARS-CoV-2 has threatened and is still threatening the lives of many. Imagine a world without biosafety, what would that be? It is going to be definitely more serious than what we are facing nowadays. So is biosafety important? Yes, that's for sure. So hazard can actually be present in any laboratory, and the types of hazards present in the lab depends on the type of work conducted in the lab. Hazard is defined as anything that is naturally dangerous or can cause harm. It can come in the form of an object, chemicals, biological agents, or even situations. These are some of the symbols often used to indicate the different types of hazard and to warn others of hazards. Hazard can be categorized into three main groups: physicals, chemicals, and biological hazard. Examples of physical hazards include electrical hazards, laser and cold hazard, sharp objects, chemical hazard including explosive materials, corrosive substances, and also flammable materials. As for biological hazards, it includes infectious agents and also infected tissues or organs. Today, we are going to focus on biohazards, which is closely related to biosafety. So what is biosafety? Biosafety is basically referring to the containment principles, technology, practices that are implemented to prevent unintentional exposures to biological agents and their inadvertent release according to WHO. Some essential components within biosafety include the following, such as biorisk assessment and identifications, specific biosafety measure, such as code of practice, physical plan, including the lab design and facility, personal protective equipment and equipment maintenance, medical surveillance and post-exposure, and also the lab worker biosafety training. Other than that, waste management program, emergency action plan, commissionings, and also certification guides for the facilities are also important components of biosafety. So why biosafety practice? Biosafety is actually very important to protect laboratory personnel and researchers from harmful incidents. It is also important to reduce the risk of accidental exposure, to protect the surroundings and environments from the biohazards such as the results of technology utilizations, and also to ensure the safe utilizations of technologies on biological agents. Therefore, compliance with regulatory standard is the key in making sure that everyone can work in a safe environment. And this is not the responsibility of one single party. This is a team effort that requires the commitment and cooperation of everyone involved in such a project, which includes the principal investigator, research lab personnel, students, and also biosafety committee as well as biosafety officer. It is the responsibility of the principal investigator to obtain approval from biosafety committee prior to commencement of work to comply with specific biosafety protocol, practices, and also procedures. And it's also the responsibility of the principal investigator to ensure all laboratory staff are appropriately trained and to report incidents and accidents of biohazards. Other than the PI, lab staff and students are also key players in determining whether or not biosafety practice are being observed in the lab. It is important to comply with the specific biosafety practice and procedures to minimize the risk of unintentional exposures to biohazard. In addition to that, the reporting of biohazard accidents or incidents is also equally important. Biosafety training is important as human error or poor technique can actually compromise the best of safeguarding the safety of laboratory workers. A safety-conscious staff or students is the key to prevention of laboratory-acquired infections incidents and also accidents. Other than that, the biosafety officers also play a very important role in making sure that biosafety procedures and policies are being followed in a particular institutions. The biosafety officers has the responsibility to conduct periodic inspections, to report any significant problems, violations, and significant research-related accidents or illnesses, to develop an emergency plan for handling accidental spills or personal contaminations, to provide technical advice to principal investigator on research safety procedures and also laboratory security. So we have been talking about hazard and we have been talking about biosafety for a bit now. So what exactly is biohazard? Biohazard material is referring to any biological substances that poses a threat to the health of living organisms. And examples of biohazard materials include body fluids and blood that contain infectious agents, infected animals, so humans' organs, tissues, or body part, microorganisms such as bacteria and fungus, viruses, prions, cell culture used in the lab, parasites and toxins, and also including allergens and toxins produced or derived from plants or animals. Other than those mentioned earlier, there are also other types of materials that are considered as biohazard, which include culture and stock, such as waste from the biological productions of life or attenuated vaccines, anything that is used to contain, to mix, to transfer agents, the biological agents, including the petri dishes, pipette tips, microtetraplates, and disposable lube. Sharps such as needles, bullets, and valves that may have been in contact with infectious materials, and also research animal waste including contaminated carcasses, body part, bedding of the animals that were exposed to infectious agents during research or testing are also included under the biohazard materials. These are some of the examples of biohazard agents that can actually be present in any research lab, mainly dealing with biological organisms, which includes infectious microorganisms, diagnostic specimens, recombinant DNA, genetically modified organisms, humans and non-human primates, cells or cell cultures, animals or plant cells or cell cultures, fluids or tissues that which may have contained pathogens, and also animal known to be a reservoir of zoonotic diseases. So these are some of the examples of biohazard agents that may be present in the lab. When we come back later on, we'll talk about what are the biosafety procedures or practice that can be used to handle these different biohazards. We're going to take a short break, and meanwhile, you can scan this QR code for your attendance. Depending on the types of biohazard, different level of biosafety is necessary, and depending on the biorisk, different facilities are required. Biosafety is categorized into 4 levels based on these biorisks. WHO classified biohazard into 4 risk groups based on the pathogenicity, mode of transmissions, host range, availability of effective preventive and treatment measure. So this is the table 1 from the WHO Lab Safety Manual 3rd editions, where it's classified the organism into 4 risk groups. So risk group 1s are referring to organisms that are microorganisms that are unlikely to cause humans or animal diseases. They pose low to no individual's cognitive risk. Risk group 2 are pathogens that can cause humans and animal diseases, but are unlikely to be serious hazards to laboratory workers, the community, livestock, and the environment. Laboratory exposures may cause serious infections, but effective treatments and also preventive measures are available, and the risk of spread of infection is limited. So these pathogens, they are in the category of moderate individual risk and also low community risk. Risk group 3 including pathogens that are usually causes serious animals and humans diseases but does not ordinarily spread from one infected individuals to another. And effective treatments and also preventive measures are also available. These pathogens pose high individual risk but low community risk. The last risk group 4, this is the group that require maximum biosafety level. These risk group 4 pathogens are pathogens that usually causes serious humans and also animal diseases and can be readily transmitted from one individual to another directly or indirectly. Effective treatments and prevention methods are usually not available in this case. So risk group 4 pathogens or agents, they pose high individuals and community risk. Examples of risk group 1 agents includes yeast, E. coli, and lactobacillus. Examples of risk group 2 pathogens includes herpes simplex and also co-viruses and also salmonella. Examples of risk group 3 includes tuberculosis, SARS-CoV-2, and also avian influenza virus. And last but not least, example for risk group 4, we have Ebola and also smallpox viruses. This table here is the information provided in the WHO Lab Biosafety Manual, where it relates the risk group to biosafety level, practice, and also equipment. So according to WHO, a biosafety level 1 lab is suitable for basic teaching and also research. Biosafety level 2 is suitable for primary health services and also diagnostic services and research. Biosafety level 3 is suitable for research where specific diagnostic services and also research are required. And last but not least, biosafety level 4 with maximum containment. This type of lab are suitable to handle dangerous pathogens and unit. This is a summary of the biosafety level requirements for the different biosafety labs, where some of the labs require isolations of laboratory and some of the labs actually required specific requirement for ventilations, double door entry, and also a personal shower room, and also autoclave and also biosafety cabinets. We're going to talk about each of these biosafety labs individually. So in this picture here, it shows an overall facilities and also the equipments that are required in a biosafety lab. The one that are numbered in orange color are those that are required in the lab, and the one that are numbered in green color are the ones that are optionals for the lab according to the needs. Different facilities are required in different labs depending on the risk of the biohazards. So for biosafety level 1, this type of lab, they are suitable for works involving agents of no knowns or minimal potential hazard, and some of the facilities required in the lab including a control access, hand washing sink, sharp hazards warning policy, personal protective equipment such as glove, lab coat, and also eye protections, and also a laboratory bench. And in this kind of lab, BSL-1 autoclave is optional depending on the type of research conducted in the lab. For BSL-2, the facilities are similar to BSL-1 with additional facility where a physical containment devices or biosafety cabinets are necessary in BSL-2. And autoclave, unlike BSL-1 which is optional, autoclave is a requirement in BSL-2. BSL-2 is suitable for work involving agents of moderate potential hazard to personals and the environment. And some of the personal protective equipments that can be worn in this lab including face shields, lab coat, and also eye protections apparatus. A BSL-3 has all the facility as in BSL-2, however with additional practice where specialized facility has to be included. A self-closing double door entry is required. airtight during disinfection, and also specific airflow, where the airflow into the lab without recirculations to non-lab areas is required. Other than that, some optional facilities such as personal shower out, power air purifying respirator, influence decontaminant systems, exhaust HEPA filter are also optional depending on the lab. This type of lab, they are suitable for work involving agents of high individual risk and low community risk. As for BSL-4, it has all the facilities required in BSL-3, however with additional practices where highly specialized facilities are required. So one of the highly specialized facilities includes the positive pressure protective suit, a chemical shower out, and personal shower out, which is optional in BSL-3, but it's actually required in BSL-4. Influence decontaminant system and also exhaust HEPA filter, which are also optional in BSL-3, they are actually required in BSL-4. This type of lab is suitable for work involving pathogens of high individuals and also community risk. One other key component of biosafety is waste management. In laboratory, decontaminants of waste and their ultimate disposals are closely related. In terms of daily use, some of the contaminations material require actual removal from the laboratory and complete destructions. And in most cases, glasses, instruments, and also laboratory clothing are normally recycled. Therefore, proper disinfections is very, very important. All infectious material should be decontaminated, autoclaved, or incinerated within the laboratory. And material for decontaminations and disposal should be placed in containers such as autoclaved plastic bags that are color-coded according to the contents to be autoclaved or incinerated. The biosafety in Malaysia is regulated under the Biosafety Act 2007. Under this act, the National Biosafety Board was established to regulate the release, importations, exportations, and content use of living modified organisms in order to protect humans, plants and animal health, the environment, and the biological diversity. The WHO Laboratory Biosafety Manual has been used broadly in all levels of clinicals and public health laboratory and also other biochemical sectors globally. It serves as a de facto gold standard, global standards that presents best practice and set trends in biosafety. In this particular biosafety manual, it includes risk assessment, core requirements, heightened core measures, maximum containment measures, transfer and transportations, and laboratory biosecurity. These are some of the sources where biosafety information may be obtained. As mentioned earlier on, the Laboratory Biosafety Manual prepared by WHO, the Biosafety Act 2007 of Malaysia, and also the Institutional Biosafety Committee, and last but not least, knowledgeable principal investigators, also lab personnel that have been properly equipped and also trained with biosafety knowledge. So biosafety in UPM is placed under the wing of UPM Institutional Biosafety and Biosecurity Committee. A laboratory biosafety document has been prepared which contains the policy and the procedures that governs all activity involving in handling, manipulating, working, using, storing and disposal of biological agents, materials including GMO, LMO, rDNA and biological toxins in defined facilities and containment zones in UPM. The procedures cover activities in all types of facilities including teaching, research, clinicals and environmental laboratory. It also covers both deliberate use of biological agents and work with material that contain or could contain biological agents. This policies or document was prepared by referring to numerous documents related to biosafety, including the Biosafety Compounding of Offenses Regulations 2018, Biosafety Sampling Procedures Regulations 2018, and also the Malaysians Laboratory Biosafety Biosecurity Policy Guidelines prepared by the Ministry of Health Malaysia 2015. Risk assessment is the backbone of biosafety. It is a continuous process to identify, to access, and also control and monitor risks. It must be accessed by professionals who have been given proper trainings and exposure. There are 5 main components involved in risk assessment process, which are to identify hazards and risks, to evaluate the risks, to determine control, to implement controls, and to review effectiveness of the control. And if the controls are not effective, then the cycle will repeat to improve the effective controls measures. Some of the factors to be considered when assessing risk including pathogenicities of the agents and infectious dose, potential outcome of exposure, natural risk of infections, stability of the agents in the environment, concentrations of the agents and volumes concentrated material to be manipulated, presence of a suitable host, and information available from animal study and reports of laboratory-acquired infections and clinical report. Laboratory activities, plans, and also genetic manipulations of organisms that may extend the host range of the agents and also alter the agent's sensitivity to non-effective treatment regimen. And last but not least, local availability of effective prophylaxis and also therapeutic interventions. So these are some of the factors that must be considered when assessing risk. These are some of the biosafety practices that I believe that everyone working in the research laboratory should adhere to, and they are mostly doable and achievable. The first one, laboratory coverall, also gown, must be worn all time for work in the laboratory. Appropriate glove must be worn for all procedures that may involve direct or accidental contact with samples such as blood, body fluids, and other potentially infectious materials or infected animals. And after use, the glove should be removed aseptically and hand must be washed. Personnel must wash their hand after handling infectious materials and animals and before they leave the laboratory. Safety glasses and face shields and other protective devices must be worn when necessary to protect the eye and face from splashes, impacting objects, and sources of artificial ultraviolet radiations. It is also prohibited to wear protective laboratory clothing outside the lab, for example in the canteen, in coffee rooms, office, library, staff rooms, and toilet. Open-toe footwear must not be worn in the laboratory. Eating, drinking, smoking, applying cosmetic, or handling contact lenses is prohibited in the laboratory working area as well. Storing human food or drink anywhere in the laboratory area is also prohibited. And last but not least, protective laboratory clothing must be used in the lab must not be stored in the SAM locker or cupboard as street clothing. With this simple biosafety practice, it is hoped that everyone would do their part to make the laboratory a safe place to work in, to minimize the accidental exposures to harmful biohazard. And that is all from me today. Hopefully the information shared today will be useful to everyone. Thank you and stay safe.